To continue with the macromolecule of protein, we also cover enzyme. We cover enzyme because not because only it is part of protein, but once we study about metabolic pathway, you will again encounter enzyme in metabolic pathways. So we start off by actually looking into some overview on enzyme. The first fact is all enzymes are protein. So that is true until they found out that there is one exception to it, where nucleic acid also can behave like enzyme. And then they enhance the rate of thermodynamically feasible reaction in living cell as well as in vitro. So the biological catalysts do not require you to raise the temperature uh, every time they need to change the substrate to product. That's the uniqueness about enzyme. In fact, in our body, the whole temperature of a person is 37 degrees Celsius, and all enzyme can perform at its very best into changing the substrate to product. Unlike the inorganic catalyst, where you need to raise the temperature every time there's a reaction up to 1000 degrees Celsius in order for the substrate to become a product. And then the third facts, enzymes are not permanently altered in the catalytic process. When the substrate is received by the enzyme, it keeps on changing it to product without it being used up or denatured or destroyed. So it's ready to continue the reaction every instance of changing substrate to product. And the fourth one, different version of the same enzyme are called isozyme or isozymes. Okay, so this one, the structure, the subunits in the enzyme may vary, but the catalyst capability only slightly different compared to the other isozyme. One example is lactate dehydrogenase. This lactate dehydrogenase in the muscle cells, myocyte, they are capable of converting pyruvate to lactic acid. But in the heart muscle, the isozymes for lactate dehydrogenase works in reverse, where it changes lactic acid to pyruvate. Why? Because you don't want the lactic acid to be so acidic that it destroys the heart muscle, and then heart is very important, right? You don't want heart to stop working. So that's the purpose of lactate dehydrogenase isozyme in the heart muscle. Then the last facts is basically already been covered, that enzyme has an active site. It receives a substrate. The substrate could be a single substrate or two substrate or even more than two, and it comes into the active site and gets converted to product. Some could be becoming two product, from single substrate become two product, or from two substrate become single bigger product. A bit on the names of enzyme. All the enzyme, most of the enzyme, and with this they call it suffix of ase. For instance, based on the substance or the substrate that they react upon, it could be, for instance, sucrose. Then it is called sucrase, and then if it's lipid, it's called lipase, and also could be because of the function of that particular enzyme. If it's oxidation that is involved, then it's called oxidase, and if it's hydrolysis, it's hydrolase. Of course, there are also some enzymes that has maintained the name as it's been referred much earlier before this ASE came about. For instance, pepsin and trypsin, which has maintained to be ending with IN rather than ASE. Next, on the classification, which has been adopted or put in place by certain association of biochemistry. So you have the classification of enzyme in these six major groups, and this is the Enzyme Commission (EC) for lactate dehydrogenase. You can see the first number one is actually referring to oxidoreductase or oxidoreductases. So lactate dehydrogenase normally involve in two-in-one reaction in receiving the substrate and causing it to be oxidized, and another reaction where the for instance, the electron will be accepted by coenzyme and therefore it will be reduced. This is normally found in pathways such as glycolytic pathway, citric acid cycle. We will see that later, where the dehydrogenase play an important role for redox reaction. Then you have number two, you have transferase. This one, for instance, protein kinases, where they are involved in adding phosphate to any particular protein, and they then will activate or inactivate the enzyme. Hydrolysis, therefore, from something big, they will cut or cleave it, such as proteases. You have lyases, where they generate double bond, and then you have isomerase, where they will convert from one isomer to the next isomer. And lastly, you will have ligases, which carries the number 6. This one creates a bigger compound by using perhaps two little compounds. So these are six categories which, if you purchase any enzyme or you study any enzyme, it normally will have this EC number with the first number happen to be either one of these six numbers, uh, basically identifying what type of enzyme it is. Then you have the subclasses that goes on. Then you have also enzymes do not function on its own, not only entirely built as protein. It also needs non-protein groups to ensure that the function is complete. For that purpose, as a whole, it is referred, the non-protein component of an enzyme is referred as cofactors, and they are then divided into categories. For instance, you have prosthetic groups. These are like, born with the enzyme itself, such as now you can see the photo there on nitrogenase. You have ferrum and molybdenum, which are actually tightly bound in the nitrogenase. So these are prosthetic group. Then you have cofactors that are bound and released whenever there is a reaction that is taking place, and these are called coenzyme. You have lots of coenzymes such as NAD, FAD, coenzyme A, biotin, pyridoxal phosphate, many of the coenzyme, and these coenzyme are actually built by vitamins. That's why vitamin B is very important in ensuring you have sufficient amount to build the coenzyme. Therefore, metabolic pathway rely on vitamin and mostly vitamin B. So here, given the list of important coenzyme that actually will play roles, important roles in metabolic pathways. So I've already highlighted some of them to be in red to identify those that are important. So basically, just now I said they are built by vitamin B, and they are useful as the non-protein portion of enzyme. They only come together when the reaction is taking place, not tightly bound, and then they are released. For instance, you have coenzyme A, which will then react with acyl group and become acetyl coenzyme A. It's a very important intermediate in the metabolic pathway, which can enter citric acid cycle or even can be used in making fatty acid, making cholesterol. Then you have flavin, which is a component of FAD, which can be reduced to become FADH2. Then once it enters electron transport chain, you can get ATP. You also have nicotinamide, component of NAD, as well as NADP. These are two coenzyme important as well. And then you have PLP, pyridoxal phosphate, also plays a role in amino acid metabolic pathway. Now we talk about the general properties of enzyme. They are having higher reaction rates. That means they can immediately take up the substrate, immediately become in the transition state, and quickly become product in a very short period of time, and readily available to do that again and again. Milder reaction condition, meaning that you do not need to raise up the temperature to have the reaction to proceed or complete. They are able to do even in a body temperature which is very low, only 37 degrees Celsius. Greater reaction specificity, meaning as you can see, the even if you have two keys for one lock, only that can fit precisely same shape with the lock only can open, while the other one cannot open the lock. And then capacity for regulation, enzyme allows itself to be regulated, to be controlled, to be inhibited, to be activated. Therefore, important in metabolic pathway. When enzyme is present in a metabolic pathway, you can therefore control the metabolic pathway by controlling this enzyme itself. So this slide proves that it has a high reaction rate enzyme, where I taken an example of carbonic acid in the body, where in the blood carbon dioxide will then dissolve with water and react and become carbonic acid, and it will then go to the lungs and then reverse the process to remove carbon dioxide from the body. And this is facilitated with the enzyme that is present, carbonic anhydrase. As you can see, based on the presence of this enzyme, and if it's not present, there's a huge difference in the numbers, where one is 10 power of 7 while the other is a mere 10 power of 5. If you think there's no difference there, please write it down in a piece of paper and you see how big of a value these two numbers are. So how do enzyme work in a milder reaction condition? To answer this, first we look at any reaction. In general, every reaction is works by collision. So when you increase the temperature slowly, the two substrate will vibrate and then bounce over each other. And the more you increase the temperature, it started to collide and causes the interaction between them. At some point, the collision will cause interactions to be rearranged. The bonds among them will be rearranged and finally create a new product or product. So that's how general reaction takes place. But with the presence of enzyme, and also the fact that in biological system you cannot be increasing temperature as you like, where organic matters may end up getting spoiled or destroyed by high temperature. Therefore, the solution is to use enzyme where it can lower the activation energy, meaning it uses a different route altogether in order to convert substrate to product without the use of very high temperature. So that's the solution that enzyme provide, that is using a different way, and it's a shortcut altogether. So this is another way to represent by diagram itself what I just explained about how enzyme can perform reaction at a low temperature. So you have here two different route. This is the route that is taken by the substrate to product without the presence of enzyme, and then you can see the route taken when there is an enzyme to convert it to product. The first route is a longer route and require more time, and the second route is shorter, therefore the reaction rate is much faster, and that's provided by enzyme. Just in order for you to not get confused about the activation energy that enzyme brings down, you can see this is the activation energy of uncatalyzed enzyme, and this one is catalyzed. You can see there is a drop in the amount when it is in the presence of enzyme. Do not get confused with this one here. This one is referring to Gibbs free energy. That is to identify whether the reaction is exothermic or endogonic. And this one is exogonic. We now move our attention to the specificity of an enzyme. Enzyme is always known to be very specific, and shown here in terms of the enzyme here with an active site, and this active site is spherical or round shape, and you can see only these to be similar to that active site, therefore it is specific compared to the other structures unable to fit into the active site. So enzyme is very specific, and in terms of specificity, it looks like there are three: absolute, relative, and also stereo specificity, which we will see in a while what is the difference from each of them. This is absolute specificity, so the structure needs to be very similar to the active site in order for it to accept and change it to become product. And that's you can see that methylurea, due to the presence of methyl, unable to fit into the active site, therefore it will not be converted to product. But urea can be, because it has the shape of the active site of urease, and therefore it can be converted to ammonia as well as carbon dioxide. Next is on relative specificity. This one got three enzymes: protein kinase A, kinase C, kinase G. All three, when they see a protein that has serine as well as threonine, they are able to phosphorylate, meaning they add phosphate. Therefore, a bit not so specific. They see any protein, all three can then put phosphate to that particular protein. Next, we have stereospecificity, and I think this is the most specific one, because just for the difference of L or D form, and therefore it's very choosing only one type of stereoisomer, and only one that will be converted to product, while the other will not be activated or converted to product at all. So highly specific is the stereospecificity. Then we have also the lock and key model, as well as later we'll see induced fit hypothesis, where it gives an idea on how the active site is capable to bind to the substrate. This was actually originally was thought of, and After a while, they realize that even though the substrate do not have the shape that is found in the active site, still it can be converted to product. Therefore, later came the induced fit model as well. And here you can see the lock and key model where the shape makes a huge difference. If the substrate has the same shape to the active site, therefore it will bind to the active site of that and only that particular enzyme and cause the transition state to come about, rearrangement of bonds to become finally product. Next is the induced fit model. Unlike the lock and key, this one looks into changing the shape of the active site only when the presence of the particular substrate come close to the active site, and therefore conformational changes takes place and it will be very much fitting the substrate. So as you can see here, there is, the active site firstly not able to accept the substrate, but as it is found to be closer, it causes a change, and then it fits, and finally products are coming about. At the bottom is actually part of actual enzyme that is hexokinase or glucokinase, they call it in the glycolytic pathway, where once glucose comes close, it changes the shape and accepts only glucose and changes it to glucose 6-phosphate. So here, given the comparison between induced feed theory as well as lock and key feed, I think it's straightforward to understand that induced feed does not have the active site shape that is required for the substrate, but once substrates come, it will then changes and fit accordingly, while the lock and key has the shape from the beginning for the substrate itself. Now we look at all the factors that can affect the enzyme action itself. We start with the temperature. So there will be no activity or little activity at low temperature, and then it increases to the highest point, and beyond that it actually becomes denatured and loses its activity. So for any particular enzyme, there should have an optimum temperature where it performs the best. Shown here, the diagram on what I just explained in the previous slide. You have it increases, collision is increased, and therefore approaching the active site is much more easier when the temperature is increased slowly, and looks like at 37 degrees Celsius, but only for matter of human. Other organism may have different temperatures when they perform the best. But for human, 37 degrees looks like the best temperature for maximum reaction rate for all the enzyme found in the body. But if say you get high fever or you become sick, that increases the temperature, denaturation could take place and therefore activity decreases for the enzyme, simply because the active site is now being distorted or denatured, as can see, and therefore the shape is no longer fitting into the denatured enzyme. Next, we look at the pH itself that can be affected if pH is changed beyond its optimum pH. Any enzyme will perform at its best at the optimum pH because the charges are acceptable between the substrate as well as the active site of the enzyme. But since there are charges and pH disturbs the charges at some instance, when you change the pH, an opposite what was say negatively charged at the active site therefore can attract the substrate which is positively charged. Some changes to the pH could cause the positive charge in the active side, therefore positive and positive does not attract anymore, so there is no activity at all between the substrate and enzyme. So here shown the what I was referring about the pH itself. There is optimum pH that the enzyme will work at its best. In fact, in the human body, unlike the one temperature for the whole range of enzyme that is found in your body to work, that is 37 degrees Celsius. For pH, you can see that different compartment in your body has a different optimum pH for the particular enzyme found that in that compartment. For instance, you have in the stomach pepsin having optimum pH of pH 2. So that's on pepsin. Then you have in the mouth cavity amylase having an optimum pH of 7 neutral. While in the liver, arginase has a optimum pH of around 10. So different pH compared to one temperature in your body to perform the best in terms of optimum. The next factor is actually substrate concentration. So you'll be surprised that you think that enzyme concentration is important. You have more enzyme, more reaction rate, but surprisingly, increasing the substrate also possible to increase the reaction rate. So what happens is more substrate, more availability of the substrate to go and find the active site of the enzyme. Therefore, reaction rate also increases. But there is a limitation where after you increase more and more substrate, at particular concentration, you will see no more increase in the reaction rate because all the substrate already found the active site and its enzyme is now saturated with substrate. Therefore, it start to plateauing in terms of the reaction rate itself. So this figure shows you what I was just explaining, where you can see as you increase the substrate, the reaction rate also increases, and once all or much substrate is available but not much enzyme is available, it will then be saturated and a plateauing effect will come about. If you want to increase further, you just have to increase the enzyme concentration, and therefore it increases like this. And again, it will reach a plateau when saturation is achieved. So these two guys, Michaelis Menten, they came up with kinetics in order to measure the activity of an enzyme, and they utilize this graph that is having initial velocity over substrate concentration. Uh, then they also derive this formula. Okay, and what's the usefulness of this formula and as well as the graph was in terms of if you have one enzyme and sorry, you have if you have two enzymes and you want to see which one works well for a particular substrate. So you do the experiment, you build this graph, and then once you build it, you find out the V max at the asymptote, they call it, and then half of that V max is referred as Km value. You do it for the both enzyme, and then the enzyme which has the lowest Km value will be the best enzyme that has better affinity for that particular substrate. So next we look at the enzyme inhibition. Inhibition can be referred in two major categories. So one is called irreversible and the other one is reversible. The irreversible causes once the inhibitor binds, it destroys the enzyme for good and therefore it's not able to perform anymore. And that's as such is called irreversible. Reversible, on the other hand, is it binds and then once it leaves the enzyme can again perform. Under the category of reversible, there are two types. One is competitive and the other one is non-competitive. The competitive one is basically looking after the active site. The inhibitor has the same shape as the active site, therefore it competes with the substrate to find the active site. If the substrate finds the active site first, therefore substrate become product, but if the inhibitor on the other hand gets the active site, no reaction takes place. The non-competitive one, unlike a competitive one, does not go to the active site, but it goes to another site that is referred as regulatory site. And if the inhibitor binds to the regulatory site, it causes inhibition altogether to the active site by causing a conformational change to the active site, which no longer can hold the substrate. So let's look at the reversible inhibitions. Firstly, with the competitive inhibitor, where, as I already explained, it looks at the active site, and if it is found the active site, therefore it inhibits. And there is one way for you to overcome this inhibition of the enzyme by simply increasing the number of substrates. So increasing concentration of substrate, meaning more substrate is available to compete with less inhibitors, and therefore maybe substrate overcomes inhibitor at a particular high concentration. Then you have the non-competitive inhibition being explained here, whereby it causes a conformational change to the active site by binding to the regulatory site instead. And unlike the competitive inhibitor, no such luck here. If you increase the concentration of substrate, it does not play any role in causing or removing the inhibition, because the site that is bound by the non-competitive inhibitor is not in the active site, is actually at the regulatory site. So again, unlike competitive inhibitor, increasing the substrate concentration has no effect on inhibition of a non-competitive inhibitor. And then something you need to know about enzyme that is found in a metabolic pathway as well. There is regulation, and it is highly regulated. And these are three ways where all the range of enzyme can be regulated. It could be regulated by just changing the enzyme quantity, and this could be done by when you don't need it, you degrade the enzyme, and when you need more of the activity, you then produce the enzyme. And this is a very slow process where it requires transcription, translation, from DNA to protein and therefore enzyme. And normally enzymes that are regulated in this way are very not the key regulatory enzyme in a pathway, but rather the common enzymes. Then a more popular way of controlling the enzyme or regulating the enzyme is actually adjusting the enzyme activity. this is what we're going to discuss in a few slides to come. But just to also talk about the other way that is changing the substrate concentration, meaning when there is a lot of the precursor or the intermediate that goes into the reaction of that particular enzyme, therefore the enzyme will be activated much more, and when there is less substrate, less the activity will be of that particular enzyme. So here we look at how to regulate the enzyme by regulating the activity alone, and that's done by regulating at the regulatory site rather than the catalytic site. So just to look at this figure to explain, you have the regulatory side as well as the catalytic side. Say you have inhibitor, and therefore it is referred as allosteric inhibition. So what happens is this inhibitor, once it binds to the regulatory side, it causes a change that looks like the catalytic side does not have the shape for the substrate, and therefore it is now inhibited. So this is an example of to explain to you what is the meaning of allosteric regulation. This allosteric regulation can be either positive or negative. So here to explain about the allosteric inhibition, you have the negative effector, where you can see at the moment it is accepting the substrate because it has the shape, but because of the inhibitor binding at this regulatory site, the shape changes and no longer able to allow the substrate to sit at the active site. Similarly, you also have the positive effector under allosteric regulation. So what was not accepting the substrate because of the shape, once the positive effector binds to this region here, it now changes the shape that has a similar shape to the substrate, and therefore the reaction can proceed. So another two ways apart from allosteric regulation in controlling the enzyme activity is covalent modification as well as association dissociation of subunits. Enzyme can also be controlled by putting a very small change or addition, and this addition or removal is only using compounds such as phosphate, which is very small compared to the entire enzyme itself, and this is done by another enzyme, that is kinases. And once the enzyme is phosphorylated, it will be either activated or deactivated depending on which enzyme that we are looking at. So that's covalent modification. Then you also have this association dissociation, and this one is using such aid or help of for instance a G protein. This one involved in the signal transduction, where the enzyme could be bound with a G protein, and therefore it also activates the enzyme entirely. So you would have noticed that I have gone through this lecture quite fast, and that is because it is not an entirely very important lecture. It's supposed to be a recap or revision for you in terms of what is enzyme, and it is also part of a bigger topic, that is protein. So I end this lecture by giving the student activities, that is on listing out differences between ribosome and also explaining the contribution of enzyme in the industry, which is relevant to healthcare, also detergent which has been incorporated to biological catalysts, leather industry in preparation of the leather, as well as also in military. Uh, this you will recap again in the next semester, where you take up enzyme technology as a course to study.